In this presentation, I will be discussing the effect of UV radiation on lice mutations in yeast. In order to provide a full scope on the results of this experiment, it is necessary that background is provided, as well as a connection to humans in everyday life. There is no way for humans to avoid the sun, and sadly the UV light that comes along with it. From the hours of 10 a.m. and 4 p.m., the UV radiation absorbed by the Earth is at its highest, and avoiding this on a daily basis is nearly impossible for most people. Alongside tanning booths, x-rays, and various other experiences also cause people to be exposed to UV. 66% of melanoma skin cancers and 90% of non-melanoma skin cancers are caused by UV exposure, which makes it imperative and necessary to create protective tools. The process is depicted in the first diagram. When UV rays from the sun hit a skin cell, the DNA often becomes damaged, usually mismatched bonding or double-stranded breaks. These mutations often lead a cell to divide uncontrollably, which could eventually lead to tumorigenesis, which can be lethal if not treated promptly and correctly. This experiment utilized the lysine synthesis pathway in yeast seen in the second diagram to quantify the rate of mutation when yeasts are exposed to 10 seconds of UV light. The step exploited in this experiment was the conversion of alpha-aminoadipate, or alpha-AA, into alpha-aminoadipate semialdehyde, or alpha-AA semialdehyde. This step is significant because reducing alpha-AA to alpha-AA semialdehyde is considered fast, but in the next step occurs slowly. If alpha-AA semialdehyde gets too high a concentration in the cell will die. This selection will allow us to isolate colonies with mutations in the lyse genes that normally function in the pathway. Lyse2 codes for the protein that reduces alpha-AA to alpha-AA semialdehyde and is activated by lyse5. So if there's a mutation in either of these genes, the cells will not be able to produce their own lysine. In order to accurately quantify the rate of mutation, a viability assay must be performed. 3 dilutions, 1 to 10 to the 4th, 1 to 10 to the 5th, and 1 to 10 to the 6th of wild-type stock yeast was plated on YPD plates in duplicates. One set was exposed to 10 seconds of UV light while the other was not. Since the plates are in complete media, both wild-type and mutant yeast will grow to determine how many cells were originally plated for further calculations. Another set of plates was also set up to create mutants. Using 2 alpha-AA plates with lysine, 1x stock yeast was plated. One plate was then exposed to 10 seconds of UV light and the other was not. Since these plates only have alpha-AA as a nitrogen source and lysine present, only mutant colonies can grow, selecting against wild-type yeast. As explained before, the wild-type yeast will die because they have too high of an alpha-AA semialdehyde concentration. Once the alpha-AA plates were finished growing, they were struck out. This means that a sterile toothpick was used to pick a mutant colony from the plate and spread across a localized area on another YPD plate to grow without any environmental stress. Once the YPD plates with the streaked colonies grew for a day, they were replica plated. This is a technique used to transfer the colonies from one plate to another using a velvet. This was done for both YPD plates, and each plate was replica plated twice, resulting in 4 plates total, 2 for the 10-second UV exposure and 2 for the no UV exposure. The colonies were replica plated onto minimal media plates, one with lysine and one without, which do not contain all nutrients necessary for growth. The MM- lice were replica plated first to avoid the risk of contaminating the plate with lysine from the other plate. The next step was a complementation assay. This means mating our unknown mutants with known mutants to identify the type of mutant we have through seeing which colonies will grow when mated. On the MM plus lice plates, all the colonies should be able to grow because since lysine is being provided, even though the colonies are mutant, they should all theoretically be able to grow. This plate acts as a control in this case, whereas the MM- plates are the test group because the plates do not contain lysine, which means that the only colonies that are able to grow are ones that have restored lyse gene function. What happens in the complementation test is that if you take a known lyse2 mutant and mate it with your unknown mutant and the colony grows at the intersection, you know it is a lyse5 mutant. You know this because the lyse2 mutant is only a lyse2 mutant, which means it still has a functional lyse5 gene. When it mates with the unknown mutant, the cells mate and become diploid, so if a colony grows, that means the diploid has a functional copy of lyse5 and lyse2 genes. We can deduce our unknown mutant must have a functional copy of lyse2 or else it would not have been able to grow, identifying that it is a lyse5 mutant. The results of these experiments provide sufficient evidence that the rate of mutation increases if yeast is exposed to UV. Since we are only analyzing the lyse genes, we cannot determine whether other genes are also being mutated, but that certainly can be the case. Additionally, there were far more lyse2 mutants than lyse5, which can lead to the next conclusion that larger genes are more likely to be mutated than smaller ones. This makes sense because if genes are larger, there's a higher probability that they will be mutated. These conclusions should be emphasized to encourage further research for protection methods to develop against UV radiation. Current methods such as sunscreen are not used widely enough, especially on a daily basis, so creating something more convenient for people to use to protect them from UV exposure is in order, and I hope that these conclusions can provide the motivation for that research. I would like to thank my teaching assistants Anna Hankin and Casey Lomar for their constant guidance through these experiments, and Dr. Lindsay Mermanesh for providing the equipment and yeast strains. Finally, thank you to the Biology Department of Brandeis University for funding this research, without which these experiments would not be possible. Thank you.